National Institute of Applied Sciences and Technology at the University of Carthage in Tunisia. And she will give now the lecture Date Palm, Pit Polyphenols, Loaded Gum, Arabic Microparticles, Influence of Egg Yolk Proteins on the Physical Properties. So Chiraz, the floor is yours.
Hello, everybody. Are you hearing me well?
Perfect.
That's fine.
Okay. I am happy to join this event with you. My name is Chiraz. I am a PhD student from Tunisia, and I am glad to present to you a part of my PhD work. My presentation is about date palm pit polyphenols, loaded gum Arabic microparticles, influence of egg yolk proteins on the physical properties. Since research is being driven by consumer needs, current increase of interest in health, food safety, quality, and freshness has pushed companies to focus on these requirements. One of most cutting-edge challenges is to replace synthetic harmful antioxidants with natural products. Polyphenols are strong candidates owing to their interesting biological activities, namely antioxidant. Researchers have investigated their biological activities and proved their important role in some chronic diseases prevention, such as cancer, diabetes, cardiovascular disease, and chronic respiratory disease. Date palm pit extract represent an interesting source of polyphenols that provide antioxidant, anti-inflammatory, antibacterial, and antiviral functions. However, the incorporation of polyphenols into food has some limitations. They can affect the organoleptic characteristic of food. They are responsible for the bitter taste, in addition to the undesired color caused by the browning reactions, besides to their high vulnerability to some environmental factors such as oxygen, light, moisture, and temperature. Polyphenols require specific conditions in food to be stable. Unfortunately, the bioavailability and the bioaccessibility of polyphenols can also be affected when they are exposed to the gastrointestinal tract. Also, polyphenols have low solubility and permeability in the gut microbiota. Thus, researchers have focused to overcome these drawbacks and found a satisfying solution. Encapsulation of phenolic content seems to be an adequate one. The capacity of the encapsulation technology to protect the bioactive compound from oxidation and degradations has pushed the food industry to use this process to enhance the development of functional food. Instead of using free compounds, polyphenols are entrapped within a carrier material. Hence, the encapsulations improves their bioavailability and maintains their stability until delivery. Moreover, encapsulation can mask the astringent taste of polyphenols, allowing their use at higher concentration for better health benefits. So, the key objectives of the work was to test the performance of date pits, polyphenols, microparticles prepared by freeze drying using gum Arabic and egg yolk protein as a novel wall material. Then to incorporate the encapsulated powders into cheese and investigate their impact on cheese texture and polyphenols content. Now we move to the material and method section, starting with microcapsule preparation. So, date palm seeds of Diglet Noor variety were purchased from Tunisian local farm. The pits were first soaked in water, washed, and then air dried and roasted at 200 degrees for 20 minutes before being ground with a heavy grinder. The extraction of polyphenols from date pits was performed using an ultrasound device. 5 grams of date pits powder was dissolved in distilled water, and the pH was adjusted to 10, which favored the liberation of the highest amount of antioxidant phenolic compounds according to preliminary testing. The amplitude of sonication was fixed at 40, the temperature at 40 degrees, and the processing time at 20. At the end of the extraction, the pH of the solution was adjusted to 7, and then they were filtered using a vacuum pump and Whatman paper. Later, we did the preparation of dilapidated egg yolk protein. And then we did the micro encapsulation by freeze drying. We prepared 5 different mixture of egg yolk proteins and gum arabic. And then samples were frozen at -80 for 12 hours, then placed into pilot scale lyophilization system for 48 hours. Samples were crushed using a mortar and pestle. In order to investigate and evaluate the microparticles, some analyses were performed, such as the moisture content, the colorimetric analysis, the scanning electron microscopy, the thermogravimetric analysis, the Fourier transform infrared spectroscopy. In addition to this analysis, antioxidant activity and encapsulation efficiency were assessed. A digestibility test was later performed. In vitro gastrointestinal simulation was studied to determine the release of apparent phenolic content by different encapsulated samples. The simulated gastric fluid and simulated intestinal fluid were prepared according to the guidelines of the US Pharmacopoeia. Results will be discussed in the next slides. This table summarizes the moisture content of freeze-dried samples. We noticed a decrease in the moisture content was observed when the extract was incorporated. This could be because protein and carbohydrates are interacting with water and polyphenols via hydrogen bonds. So the incorporation of polyphenols leaves fewer free groups available to bond water. Furthermore, the moisture content rose significantly when egg yolk protein was incorporated into the samples. When it's increasing in proportion, which suggests that the protein possesses a higher capacity to bind water than shown by the gum arabic. The table shows the results for the color parameter 2. Clearly, the addition of the date pit extract significantly changed the color of the samples, and the microparticles became darker. Taken into consideration the appearance of the raw date pit extract, this change in the color of the microparticles was expected. The successful encapsulation method relies on achieving high retention of the core material and minimum amount of the core materials on the surface of powder particles. The results obtained indicate that the type of wall materials had a significant effect on the micro encapsulation efficiency. The encapsulation efficiency ranged from 44 to 99. Clearly, the lowest encapsulation efficiency value was found when only gum arabic was used as a coating material. And in fact, the encapsulation efficiency values decrease with the increase of gum arabic concentration. The enhancing effect of the egg yolk protein on the encapsulation efficiency may be produced due to the formation of stronger interaction between the protein and the polyphenols that those products produced between the gum arabic and the polyphenols. Regarding the antioxidant activity, the wall materials used in encapsulation in the study have an antioxidant activity themselves, as is shown in the figure. When date pit extract was added, the antioxidant activity of the microparticles was significantly improved. However, the antioxidant activity of the loaded microparticles shown in the figure remained lower compared to the theoretical antioxidant activity expected, in particular when the wall material composition was high in egg yolk protein. It has been suggested that the use of protein may increase the hardness of the lyophilized cake, it being therefore more difficult to crush it into microparticles, which could lead to higher degradation of some polyphenols because of the oxidation reactions produced during the grinding of the samples. The reason may also be associated with the possible formation of strong bonds between the egg yolk protein and the phenolic compounds. And although the microparticles were totally solubilized before this assay was performed, a fraction of the proteins could be forming complexes with fraction of the phenol compounds, which would decrease the antioxidant activity. Therefore, the microparticles containing a higher amount of egg yolk protein showed better antioxidant activity. This explained by the higher antioxidant activity of egg yolk protein compared with that of the gum arabic. Micrographs of the sample are showing in the present figure. All freeze-dried encapsulated powders contain irregular shaped particles and flaky structure. These are characteristic of microparticles produced by lyophilization. In fact, during the drying process, sublimation of ice occurred, leading to a porous structure which provides more rigidity. Thus, lyophilization is considered as the best way to encapsulate bioactive compounds. This is the figure of the Fourier transform infrared spectroscopy. FTIR results for the three matrices of gum arabic and egg yolk protein at different ratios without and with date with extract and encapsulated powders show similar behavior as the previous samples depending on the percentage of the wall materials. Overall, the shift in peak positions confirms the interaction between the date with extract and the coating materials. This is the thermogravimetric analysis. We noticed that the onset temperature increased from 228 to 280 as the amount of egg yolk protein in the capsules increased. The encapsulated powder samples have better thermal stability compared to the free date pit extract. The degradation of the date pit extract started at a lower temperature at 140 than that of any encapsulated sample.
Chiraz,
therefore,
can you come to an end within 3 minutes? Is it possible?
In 3 minutes?
Yeah.
Yes, it's possible.
Okay,
I almost
great.
finished. Yeah.
Thank you.
So from this test, we conclude that the egg yolk protein and gum arabic have successfully improved the thermal stability when they were used as coating material for date pit extract. For this in vitro gastric and intestinal release of the microencapsules under simulated conditions, clearly the use of gum arabic alone as coating material allowed an important release of polyphenols from encapsulated after gastrointestinal digestion. This unexpected result is probably not related to enzyme action, but to the high solubility of gum arabic in water compared to the other gums. However, the release of polyphenols was significantly lower in the other samples and decreases with the increase in proportion of in the encapsulated powder. For the conclusion, we conclude this work with the addition of egg yolk protein raised the thermal stability of the microparticles more than did gum arabic. Encapsulation efficiency was better than egg yolk protein made up a higher proportion of the wall material. The antioxidant activity of the microparticles increased when egg yolk protein was used. In fact, egg yolk protein has antioxidant properties which further enhance the antioxidant activity of the microparticles. After in vitro digestion, the release of phenolic content in the gastric digestion was seen to be higher than the intestinal digestion, but the incorporation of egg yolk protein limited the loss of polyphenols in the gastrointestinal tract. As a perspective, we try to incorporate this encapsulated powder to food matrix, and we choose the cheese. So then when we added the encapsulated powder to cheese, we did a textural characterization of functional cheese and the digestibility test. As preliminary test, the cheese prepared maintained their textural properties, and the microparticles with a high egg yolk protein content remained undissolved in the food piece, and more analyses are needed to investigate the cheese quality. These results are published in a paper in Food and Science Technology. You can see it for further details if you want. And thank you for your attention.
Okay,
do you have
very
any
much.
Questions?
Yeah, for this very nice lecture, and the lecture is open for discussion. Are there any questions?